Welcome back to the deep dive. Today, we're really aiming high. We want to give you the ultimate shortcut to, well, professional level mastery of the gastrointestinal tract.
Yeah, we're moving beyond just simple diagrams here.
Definitely. We're decoding what's essentially the most complex chemical factory in the human body. It's this incredibly efficient absorption machine, but it's also, critically, our main interface with the outside world. Highly regulated, too.
Exactly. And our mission today, based on some really detailed source material histology, cellular function, is to dig deep. We want to pull out every key insight you need to understand this system like an elite professional.
We're talking complete mastery, down to the individual cells, how they work, where they sit.
That's the goal.
Okay, so let's kick things off with the basics. What exactly is the digestive system? We're talking about that continuous tube, right? The GI tract or alimentary canal.
Right, from the oral cavity, down the esophagus, stomach, small and large intestines, all the way to the anus. But you absolutely can't forget the associated organs.
The chemical firepower, as you put it.
Precisely. The salivary glands, the liver, the pancreas, they're essential partners in this whole process.
Functionally, it seems like it's got this dual mandate, almost contradictory.
It really does. The obvious one is getting those macromolecules, proteins, carbs, fats, nucleic acids, breaking them down into absorbable bits for energy growth maintenance.
Standard digestion and absorption.
But the second job, arguably more complex, is creating this highly selective protective barrier. Think about it, the stuff inside the lumen, the food, the bacteria, the acid. Technically, it's outside your body's internal environment.
Huh. Like an external environment running through you.
Kinda, yeah. So the tract has to be this perfect gatekeeper, deciding what gets absorbed into the body proper and what absolutely must stay out.
And it achieves this through a sequence of processes built into the structures, about 8 key ones.
That sounds right. It starts with ingestion, obviously, getting food in, then mastication, chewing it up mechanically.
Then the system takes over.
Right. Motility to move things along. Secretion, adding mucus for protection, enzymes for digestion, fluids, bile from the liver. Then there's local hormone release for signaling.
Followed by the main chemical work, digestion.
And then the crucial step, absorption of those small molecules, water, electrolytes. And finally, you have to deal with the leftovers elimination.
Okay, let's unpack the universal blueprint for this whole system then. Because from the esophagus right down to the rectum, this hollow tube generally follows the same basic 4-layer construction plan. Understanding this is, well, fundamental before we get into the regional specialization.
Absolutely. This underlying structure provides the context for all the variations we're going to see. So we start from the inside, the lumen, and work our way outwards.
Okay, layer 1, facing the food. That's the mucosa, also called the mucous membrane. But it's more complex than just a lining, isn't it?
Oh, much more. It has 3 parts. First, the epithelial lining itself. And this changes dramatically depending on where you are in the tract.
We'll definitely get into those changes.
Underneath that epithelium is the lamina propria. Think of it as loose connective tissue. It's packed with blood vessels, lymphatic vessels, immune cells like lymphocytes, and sometimes small glands.
And the third part of the mucosa.
That's the muscularis mucosae. It's a very thin layer of smooth muscle. Its job is to allow the mucosa itself to kind of twitch and fold locally. Changes the surface subtly.
Interesting. Okay, moving outwards from the mucosa.
Layer 2. The submucosa. This is a denser layer of connective tissue providing more structural support. You find larger blood vessels and lymph vessels here.
And sometimes glands or immune tissue?
Exactly. Glands can be embedded here, and significant clusters of lymphoid tissue are common. But critically, this layer houses the first of the major nerve networks we need to know, Meissner's plexus, the submucosal plexus, part of the autonomic nervous system.
Got it. Submucosa holds Meissner's plexus. Now, layer 3 must be the main muscle layer.
That's right. The muscularis, often called the muscularis externa, to distinguish it from the thin muscularis mucosae. This is the real engine for moving food along. It's thick, powerful, smooth muscle.
And typically in 2 layers.
Usually, yes. An intercircular layer where the muscle fibers wrap around the tube, and an external longitudinal layer where the fibers run along the length of the tube.
And nestled between these 2 muscle layers, another nerve plexus.
The second crucial one, Auerbach's plexus, also called the myenteric plexus. This is like the heavy-duty control system. It's made of autonomic neurons grouped into little ganglia.
And this one controls the big movements.
Precisely. The coordinated, powerful contractions driven by Auerbach's plexus are what mix the contents and propel them forward. That's peristalsis primarily.
So if you put Meissner's plexus in the submucosa and Auerbach's plexus in the muscularis together, that's the gut's own nervous system.
That's the enteric nervous system, sometimes called the second brain. And understanding this synthesis is vital for mastery. It's remarkably autonomous.
And when it goes wrong?
The consequences are severe. In conditions like Hirschsprung disease, where babies are born missing these ganglia and parts of the colon, or in infections like Chagas disease that damage the nerves, well, the tract loses coordinated motility.
Things just stop moving.
Effectively, yes.
Yeah.
Contents back up, leading to massive dilations upstream of the affected area. It also helps explain why emotions, stress, can have such a direct impact on your gut function. The nervous system connections are incredibly intimate.
Okay, final layer, layer 4. The outer wrapping. This one depends on location, right? Serosa versus adventitia.
Exactly. If the organ is hanging freely in the abdominal cavity, like most of the intestines, its outer coat is the serosa. That's a thin layer of loose connective tissue covered by a simple squamous epithelium called the mesothelium. It's slippery, allows movement.
And it connects to the mesentery.
Right, it's continuous with the mesentery that suspends the intestines.
Yeah.
But if a part of the tract is bound directly to surrounding structures, like most of the esophagus and the chest,
Then it needs anchoring, not slipperiness.
precisely, so it has an adventitia. This is a thicker layer of fibrous connective tissue that just blends into the surrounding tissues, holding it in place. No mesothelium covering.
Before we dive into specific regions, we absolutely have to talk about the immune system's presence here. MALT.
MALT, mucosa-associated lymphoid tissue. Non-negotiable for mastery. Remember that lumen barrier. It's facing thousands of microbial species.
Good guys and bad guys, presumably.
Both. So MALT, which is dense in the lamina propria and sometimes the submucosa, is the essential immunological guard force. The lamina propria in particular is loaded with macrophages and lymphocytes.
And many of those lymphocytes are making a specific antibody.
Yes, focused on producing IgA antibodies. This is where functional histology gets really cool. How do you get these antibodies out into the hostile lumen without them being instantly digested?
Good question. Passively diffused out.
No, it's an active process using the epithelial cells themselves. It's called transcytosis. The IgA binds to a receptor on the basal side of the epithelial cell, gets pulled through the cell in a vesicle.
Like an internal transport system.
Exactly. And then it's released on the apical side, into the lumen. Crucially, it's released still attached to a piece of the receptor, the secretory component.
And that protects it.
Yes. That secretory component shields the IgA from proteolytic enzymes in the lumen. It's like sending out armored antibodies directly onto the battlefield to flag bacteria and viruses before they can even try to invade.
All right, let's shift focus right to the beginning. The entrance. The oral cavity. The linings here tell a fascinating story about mechanical stress and sensory needs. It's generally stratified squamous epithelium, but the key difference is keratin, right?
Absolutely. It varies. Where you have the most friction and force, you need the toughest protection. So the masticatory mucosa, that's the gingiva, gums, and the hard palate.
Where you're grinding food.
Exactly. That mucosa is heavily keratinized, super tough surface. And uniquely, its lamina propria attaches directly to the underlying bone periosteum, very firm.
But other areas are softer.
Right. The lining mucosa covers the soft palate, the inside of the cheeks, the floor of the mouth, the pharynx. These areas need more flexibility, less abrasion resistance, so they're mostly non-keratinized.
And that flexibility allows for other structures underneath.
Yes, the underlying submucosa in these areas is thicker and often contains numerous minor salivary glands for continuous lubrication, plus diffuse lymphoid tissue for defense.
Let's talk about the lip, the labia, especially that distinct red part.
The red vermilion zone. It's a really interesting transition zone. The lip itself is highly mobile because it has a core of striated muscle. The vermilion zone has this very thin layer of keratinized epithelium.
Why is it red then?
It lacks the glands, both sweat and salivary glands, that you find on the outer skin or inner mucosal surface. This makes it prone to drying out. The pink red color comes from the underlying connective tissue being incredibly rich in capillaries and sensory nerve endings, which are visible through that thin epithelium.
And that thinness and rich innervation make it vulnerable.
Exactly. That's why viral infections like herpes simplex 1 can cause those characteristic painful lesions, cold sores, or canker sores. The virus damages the thin epithelium, flares up when immunity is low.
Okay, moving inside to the tongue. A muscular powerhouse.
Incredibly mobile, striated muscle fibers running in all directions. The top surface, the dorsal surface, is unique. You've got the sulcus terminalis, that V-shaped groove separating the front two-thirds from the back third.
And the front part is covered in bumps, papillae.
Right, 4 types of lingual papillae. Knowing these is key for mastery.
Let's break them down. Type 1.
Filiform papillae. These are the most numerous. They're conical, pointy, and heavily keratinized, which gives the tongue that whitish appearance sometimes. Their job is purely mechanical friction for moving food around. Critically, they do not have taste buds.
Okay, mechanical only. Number 2.
Fungiform papillae. Mushroom-shaped, as the name suggests, lightly keratinized, scattered among the filiform papillae. These do have taste buds, usually on their top surface.
Got it. Number 3.
Foliate papillae. These are parallel ridges found on the sides of the tongue. They have taste buds, but in adult humans, they're often sort of rudimentary, less developed.
And the last type, the big ones.
Velate papilla, or circumvelate. Much larger, maybe 1 to 3 millimeters across. There are only about 8 to 12 of them, arranged in that V shape, just in front of the sulcus terminalis. They're distinct because they're surrounded by a deep circular groove, like a moat.
A moat. Why the moat?
This is functionally important. Ducts from special serous salivary glands called von Ebner's glands empty directly into that moat.
Be continuously flushing it.
Continuously.
Yeah.
This flow of watery, enzyme-containing fluid washes away old food particles and dissolves chemicals. It keeps the taste buds located on the sides of the velate papillae, down in that moat, ready to sense new tastes rapidly. The enzymes, including lipase, also prevent oily films from blocking the receptors. Sensory optimization.
Speaking of taste, let's look at the taste buds themselves, these little onion-shaped structures.
Ovoid structures, yeah. Each contains maybe 50 to 100 cells.
Yeah.
You've got the actual gustatory taste cells, supporting cells, and basal stem cells for regeneration. They turn over pretty quickly, every 7 to 10 days.
And how do they detect taste?
The gustatory cells have microvilli at their tip that poke through an opening called the taste pore. These microvilli make contact with tastance chemicals dissolved in the saliva.
And we recognize 5 basic tastes.
Currently, yes. Salty, sour, sweet, bitter, and umami, savory. The detection mechanism differs.
How so?
Salty, sensing sodium ions, and sour, sensing hydrogen ions, protons from acids, work through direct ion channels. The ions flow in and directly change the cell's electrical potential.
Simple enough. What about the others?
Sweet, bitter, often associated with potential toxins like alkaloids, and umami, detecting amino acids like glutamate and africate, rely on more complex G-protein-coupled receptors, GPCRs.
Like many hormone receptors.
Similar principle. Binding the tastant triggers a cascade inside the cell, ultimately leading to depolarization and signaling to the associated sensory nerves. But remember, our actual perception of flavor is much richer. It integrates smell, temperature, texture, everything. Okay, now let's tackle the teeth and their support system, the periodontium. We're talking about some of the hardest, most specialized tissues in the body. 32 permanent teeth in adults, replacing the 20 baby teeth.
Each tooth has that basic structure, crown above the gum, root below, connected at the neck, and the main components are
enamel covering the crown, cementum covering the root, the bulk of the tooth made of dentin, and the central pulp cavity filled with connective tissue, nerves, and blood vessels.
Let's focus on dentin. It's harder than bone.
Yes, significantly. About 70% calcium hydroxyapatite plus type I collagen and other matrix proteins. It's produced by specialized cells called odontoblasts, which line the inner surface adjacent to the pulp cavity.
And these odontoblasts are interesting because
they persist throughout life. They're derived from neural crest cells, and they continuously lay down a layer of unmineralized matrix called predentin, which then calcifies. This allows for slow, lifelong dentin deposition, repair, and maintenance.
And they have extensions into the dentin.
Yes, long cytoplasmic processes called odontoblast. Processes extend outwards from the cell body, running through tiny channels called dentinal tubules that span the entire thickness of the dentin.
And that's related to tooth sensitivity.
Absolutely. Changes in temperature, pressure, or pH can affect the fluid within these tubules. That movement is detected by nerve endings near the pulp, often associated with the odontoblast processes, and perceived by the brain as pain.
Okay, now for the superstar. Enamel, the hardest substance in the body. What makes it so tough?
It's almost entirely mineral. We're talking 96% calcium hydroxyapatite, very little organic material, only 2–3%, and critically, no collagen. It's also cellular in its mature state.
How is it structured then?
It's composed of highly organized interlocking columns or prisms called enamel rods. Each rod runs from the dentin enamel junction to the tooth surface. This dense crystalline structure provides incredible hardness and resistance to the pressures of chewing.
But the development is key here, the cells that make enamel.
The ameloblasts. These are epithelial cells derived from ectoderm that function only during tooth development within the embryonic enamel organ. They secrete the enamel matrix proteins like amelogenin, which guide the rapid mineralization process.
And the crucial fact for mastery.
The ameloblasts are completely lost when the tooth erupts into the oral cavity. They degenerate and disappear,
Meaning?
meaning enamel cannot be regenerated or repaired by the body's own cells. Once it's formed, that's it. Damage from decay or wear is permanent. This is why fluoride is so important. It gets incorporated during development or topically to form fluorapatite, which is more resistant to acid dissolution. It's prevention, not repair.
A stark contrast to the lifelong activity of odontoblasts making dentin.
Exactly. A fundamental difference.
Okay, let's look at the periodontium, the structures that hold the tooth in place.
Right. This includes the cementum, the periodontal ligament, the alveolar bone it sits in, and the gingiva surrounding it.
Cementum covers the root.
Yes, it's a thin layer of bone-like calcified tissue, but it's avascular. It contains cells called cementocytes within lacunae, similar to bone. It anchors the periodontal ligament fibers and can undergo slow remodeling.
And the periodontal ligament, PDL?
This is critical. It's a specialized fibrous connective tissue that connects the cementum of the root to the alveolar bone socket. It's composed of bundles of collagen fibers, often called Sharpie's fibers, that embed in both cementum and bone.
So it's like a shock absorber.
In a way, yes. It allows for slight tooth movement during chewing, cushioning the forces. It's highly cellular, well-vascularized, and richly innervated, providing sensory feedback and nutrition. It also acts as the periosteum for the alveolar bone socket.
And the alveolar bone itself.
Right. It's the part of the jawbone that forms the socket. It's unique because it relies on the PDL for its periosteum and undergoes constant remodeling in response to stress.
Finally, the gingiva, the gums.
This is the specialized oral mucosa, the keratinized masticatory type we talked about, that attaches firmly to the periosteum of the alveolar bone.
And there's that little groove where the gum meets the tooth.
The gingival sulcus. A shallow groove, ideally only up to 3 millimeters deep. At the base of this sulcus is the junctional epithelium.
What's special about that?
This epithelium forms a crucial seal, attaching the gingiva to the tooth enamel surface via hemidesmosomes and a specialized basal lamina called the epithelial attachment or cuticle. The seal is the body's first line of defense against bacteria invading the periodontium.
Which is why gum disease starts here.
Exactly. Bacterial plaque buildup in the sulcus leads to inflammation gingivitis. If untreated, it progresses to periodontitis, where the inflammation destroys the junctional epithelium and the periodontal ligament fibers, leading to bone loss and eventual tooth loosening.
Okay, moving down from the oral cavity, we hit the esophagus. Basically, a muscular tube about 25 centimeters long, just for transport, right? Pharynx to stomach.
Purely transport. Structurally, it needs to handle swallowed food, which can be abrasive, so the mucosal lining remains protective. Non-keratinized, stratified squamous epithelium continues with the pharynx.
Protection also comes from mucus.
Definitely. There are small esophageal glands scattered in the submucosa throughout its length, secreting mucus for lubrication. And near the stomach junction, you find esophageal cardiac glands and the lamina propria, adding more protective mucus.
Now, the muscularis layer here has that interesting transition. It relates to swallowing, doesn't it?
Perfectly. Swallowing starts voluntarily, but finishes involuntarily via peristalsis. The muscle composition reflects this. The upper one-third of the muscularis externa is entirely skeletal muscle.
Under conscious control.
Right. The middle third is a mixture of skeletal and smooth muscle fibers, and the lower one-third is purely smooth muscle, driving involuntary peristalsis. A beautiful functional gradient.
And the outer layer. Mostly anchored.
Mostly. For most of its length in the thorax, it's covered by adventitia, blending with surrounding tissues. Only the very short segment, maybe the last 1 to 2 centimeter after it passes through the diaphragm into the abdominal cavity, is covered by serosa.
And that junction with the stomach, the lower esophageal sphincter area, is prone to issues.
Very common ones. If that sphincter isn't fully competent, acidic stomach contents can reflux back up into the esophagus. That causes heartburn or reflux esophagitis, GERD.
And long-term reflux is dangerous.
It can be. Chronic acid exposure can damage the normal squamous lining and cause it to change metaplasia into a simple columnar epithelium, resembling the stomach or intestinal lining. This condition is called Barrett esophagus, and it significantly increases the risk of esophageal adenocarcinoma.
All right, passing that sphincter, we arrive at the stomach, a J-shaped, dilated part of the tract where things get serious chemically. What are the main jobs here?
Several key things happen. It continues carbohydrate digestion, started by salivary amylase. It adds acidic fluid to the food, turning it into a slurry called chyme. It starts protein digestion using pepsin, and it begins triglyceride digestion using gastric lipase. It also acts as a reservoir.
Anatomically, we divide it into regions.
Yes, the cardia near the esophagus, the large fundus and body, which are the main sites of acid and enzyme secretion, and the pylorus, the funnel-shaped region leading to the small intestine.
And those folds inside, the rugae?
Rugae, exactly. These are large, temporary, longitudinal folds of both the mucosa and submucosa that allow the stomach to expand significantly when filled with food. They flatten out as it stretches.
Histologically, the lining makes a dramatic switch at the cardia, right?
An immediate abrupt change. The stratified squamous epithelium of the esophagus stops, and simple columnar epithelium begins. This columnar epithelium lines the entire stomach surface.
And it's not flat, it forms pits and glands.
Right. The surface invaginates to form millions of tiny openings called gastric pits. These pits are the entryways to the underlying gastric glands, which are long, branched, tubular glands extending down through the lamina propria.
What about the cells lining the surface and pits? Are they secretory?
Yes, these are surface mucus cells. Their primary job is protection. They secrete a very thick, viscous, alkaline mucus layer that's rich in bicarbonate.
To neutralize the acid.
Exactly. It forms a physical and chemical barrier, trapping bicarbonate ions right at the cell surface to neutralize the harsh HCl produced deeper in the glands before it can damage the lining. These surface cells live fast and die young dural. Is only about 4 to 7 days.
Such rapid turnover requires a stem cell source. Where are they?
They reside in a narrow region partway down the gastric pit, near where it joins the gland proper. This area is called the isthmus or neck region. These are pluripotent stem cells.
And they produce all the other cell types.
Yes. They divide, and their daughter cells migrate either upwards to replace the surface mucus cells or downwards into the gland to differentiate into the various specialized secretory cells of the gastric gland itself. A continuous renewal process.
Okay, this is where the real chemical action happens, deep in those gastric glands, especially in the fundus and body. We need to master the 4 main secretory cell types found here.
Absolutely. Their location within the gland often gives clues to their function. Let's start near the top, in the neck region.
Okay, type 1.
Mucous neck cells. They're interspersed among the other cells in the gland neck. They look a bit different from the surface mucus cells, less columnar, more rounded nucleus. They also secrete mucus, but it's less alkaline, more soluble than the thick surface mucus. Its exact role is debated, maybe lubricating gland contents.
All right, moving deeper into the gland, this next one sounds important. Parietal cells, also called oxyntic cells.
Immensely important and visually striking. They're large, often sort of pyramidal or rounded, and they stain intensely pink or red with standard H and E stains, very eosinophilic.
Why so pink?
Mitochondria. Their cytoplasm is absolutely packed with mitochondria. This reflects their huge energy demand.
Because they make
2 critical things. Hydrochloric acid, HCl, responsible for the stomach's low pH, and intrinsic factor, a glycoprotein essential for vitamin B12 absorption later on.
Let's really unpack that HCl production. How does a cell pump out such strong acid without digesting itself?
It's an amazing feat of cellular machinery, fueled by all those mitochondria. Inside the parietal cell cytoplasm, the enzyme carbonic anhydrase combines water and CO2 to make carbonic acid, which quickly dissociates into bicarbonate ions, text HgO3 ions, and hydrogen ions, textile plus.
So the cell generates the protons internally.
Yes. Then a powerful proton pump, plus text AGPase located in the cell membrane, actively pumps these text indolars out of the cell into the gland lumen in exchange for potassium ions. Chloride ions follow the protons out through separate channels, and they combine in the lumen to form HCl.
And how does the cell manage the surface area for all this pumping?
It has a specialized structure. When stimulated, internal membranes fuse to form a deep infolding of the apical membrane called the intracellular canaliculus. This dramatically increases the surface area available for the proton pumps.
What happens to the bicarbonate produced?
The balancing act. The text HTO3 is transported out of the basal side of the parietal cell into the lamina propria connective tissue in exchange for chloride ions entering the cell. This bicarbonate helps buffer the tissue fluid, contributing to that protective alkaline layer near the surface epithelium. Elegant system.
And you mentioned intrinsic factor. If that fails?
A big trouble, but downstream. Intrinsic factor binds to vitamin B12 in the stomach and protects it. This complex is then absorbed way down in the ileum. Without intrinsic factor, you can't absorb B12 efficiently.
Leading to
Pernicious anemia. B12 is vital for DNA synthesis and red blood cell production. Lack of B12 impairs the proliferation of erythroblasts in the bone marrow, causing anemia with large, fragile red cells.
Okay, parietal cells are complex. What's the third major cell type, found deeper in the glands?
These are the chief cells, also called zymogenic cells. They're typically found in the lower half or base of the gastric glands.
And they look like typical protein factories.
Exactly. They have abundant rough endoplasmic reticulum, RER, for protein synthesis, a prominent Golgi apparatus for packaging, and numerous apical secretory granules containing inactive enzyme precursors,
Precursors like
primarily pepsinogens. These are released into the acidic lumen, where the HCl cleaves off a segment, converting them into active pepsins. Pepsins are proteases. They begin the breakdown of proteins into smaller peptides. Optimal pH is very low, around 1.8 to 3.5.
do they make anything else?
Yes, they also produce gastric lipase, which starts the digestion of dietary fats, particularly triglycerides.
Okay, that's 3. The fourth type is scattered around. The hormonal communicators.
Right, the enteroendocrine cells, part of the diffuse neuroendocrine system, DNES. These are individual cells scattered mainly near the base of the glands.
And they release signals.
Yes, they function like endocrine or paracrine cells, releasing various peptide hormones or signaling molecules into the underlying lamina propria. These signals regulate things like gut motility, secretion of acid or enzymes, feelings of satiety, etc.
Do they sense the lumen contents?
Some do, some don't. There are open-type enteroendocrine cells that have a tuft of microvilli reaching the lumen, allowing them to directly sample the chemical environment. Others are closed-type, relying on other signals. A classic example is the G cell, found mainly in the pyloric glands, which releases the hormone gastrin to stimulate acid secretion by parietal cells.
And these cells can form tumors.
They can. Tumors arising from enteroendocrine cells are called carcinoid tumors. Depending on the cell type and the hormone they overproduce, like serotonin from EC cells, these can cause a range of symptoms related to excessive gut motility, vasoconstriction, or other hormonal effects.
Briefly, what about the glands in the cardia and pylorus regions? Are they the same?
Structurally similar long pits, coiled glands but functionally different. They mostly lack parietal and chief cells. Their main job is producing abundant mucus, providing lubrication and protection near the entry and exit points of the stomach. They also secrete lysozyme, an antibacterial enzyme.
And finally, the stomach's muscularis layer. Any specializations?
It's typically described as having 3 layers, though they can be indistinct. An inner oblique layer, a middle circular layer, and an outer longitudinal layer. This extra oblique layer helps with the churning motion of the stomach.
And at the exit.
The middle circular layer becomes massively thickened at the junction with the duodenum to form the powerful pyloric sphincter. This muscular ring acts like a valve, carefully controlling the emptying of acidic chyme from the stomach into the small intestine. Leaving the stomach through the pyloric sphincter, we enter the longest part of the GI tract, the small intestine, roughly 5 meters of it, divided into the duodenum, jejunum, and ileum. This is where digestion is truly completed and where the vast majority of nutrient absorption takes place.
And to do that efficiently, it employs an incredible strategy to maximize its internal surface area. It's not just a smooth tube inside.
Not at all. It's a masterclass in biological engineering, using folds upon folds upon folds. It's a geometric progression.
Okay, level 1 of folding.
Level 1 are the large, permanent, shelf-like folds of both the mucosa and submucosa that spiral around the lumen. These are the plicae circulares, or valves of Kerckering. They are most prominent in the jejunum, and they increase the surface area by about 3 times.
3 times already. Level 2.
Covering the surface of the plicae and the entire inner lining are millions of tiny, finger-like or leaf-like projections of the mucosa only. These are the villi. Each villus is 0.5 to 1.5 millimeters long. They increase the surface area by another 10 times.
Wow. So we're at 30 times the area of a smooth tube already, and each villus has a core.
Yes, a core of lamina propria containing a dense network of blood capillaries and, crucially, a single blind-ended lymphatic capillary called a lacteal. This is key for fat absorption.
Okay, that's the villi. There's a third level, microscopic.
The final magnification. The individual absorptive cells lining the villi, the enterocytes, have their own apical surface densely packed with microscopic projections called microvilli. These form what's called the striated border, or brush border.
How much do the microvilli add?
They increase the surface area by another 20 times. Each cell can have thousands of microvilli.
So 3 times, times 10 times, times 20 times, that's a 600-fold increase.
Roughly, yes. The total estimated absorptive surface area of the human small intestine is staggering, often cited as being over 200 square meters.
200 square meters, that's like a singles tennis court folded inside you. Astonishing.
It highlights the absolute necessity of efficient nutrient extraction.
Between the bases of these villi, there are glands.
Yes, the epithelium dips down to form short tubular glands called the intestinal glands, or crypts of Lieberkühn. These crypts extend down to the muscularis mucosae. And critically, the base of these crypts is where the intestinal stem cells reside.
Regenerating the entire lining.
Constantly. The entire epithelial lining of the small intestine is replaced every few days. Cells are born in the crypt, differentiate as they migrate up the villus, function at the top, and are then shed into the lumen.
Okay, for mastery, we need the specific cell types working together in this lining. There are 5 main ones.
5 key players. The most abundant, obviously, are the enterocytes, the absorptive cells.
What do they look like?
Tall columnar cells with that apical brush border of microvilli. Their main job is to absorb the final products of digestion. Embedded within their microvilli membrane and the overlying glycocalyx, a sugar coat, are the final digestive enzymes.
Like enzymes for sugars and peptides.
Exactly. Disaccharadases, like lactase, sucrose, and peptidases, break down small sugars and peptides right at the cell surface. The resulting monosaccharides and amino acids are then actively transported into the enterocyte.
And where do they go from there?
These water-soluble nutrients, sugars, amino acids, small fatty acids, are released from the basal side of the enterocyte directly into the blood capillaries within the villus core. They travel via the portal vein straight to the liver for processing.
But fats are different. You mentioned the lacteal.
Yes, lipid absorption is a specialized pathway. Larger fats, long-chain fatty acids, monoglycerides, are digested, then emulsified by bile salts from the liver into tiny droplets called micelles. These micelles diffuse to the enterocyte surface.
And the fats enter the cell.
The lipid components diffuse into the enterocyte.
Mm-hmm.
Inside the cell, in the smooth endoplasmic reticulum, they are reesterified back into triglycerides. Then, in the Golgi apparatus, these triglycerides are complexed with specific proteins, apoproteins, and packaged into relatively large lipid droplets called chylomicrons.
And these chylomicrons are too big for the blood capillaries.
Generally, yes. They are instead exocytosed from the basolateral surface of the enterocyte and enter the central lacteal, the lymphatic capillary. They travel through the lymphatic system, bypassing the liver initially, and eventually enter the bloodstream via the thoracic duct.
Fascinatingly different pathways based on solubility. Okay, second cell type.
Goblet cells, interspersed among the enterocytes, becoming more numerous as you move down the intestine. Their job is simple. Synthesize and secrete mucins, which hydrate to form mucus, providing lubrication and protection.
Third type, found deep in the crypts.
Paneth cells, located right at the base of the crypts of Lieberkühn. They are easily recognizable by their large, intensely eosinophilic, pink-staining apical granules.
And they are defenders.
Key players in innate immunity. Their granules contain antimicrobial substances like lysozyme, breaks down bacterial walls, phospholipase A2, and potent peptide antibiotics called defensins. They help regulate the microbial flora in the gut lumen, especially protecting the vital stem cell niche at the crypt base.
Fourth type, the signalers again.
Enteroendocrine cells, similar to those in the stomach, scattered mostly in the crypts. Many are the open type, sampling luminal contents and releasing hormones like secretin, cholecystokinin, CCK, GIP, which regulate pancreatic secretion, bile release, gastric emptying, and satiety.
And the fifth type is specialized for immune interaction.
Yes, the M microfold cells. These are specialized epithelial cells found primarily in the ileum, specifically overlying the large lymphoid aggregates known as Peyer's patches.
What do M cells do?
They have broad microfolds on their surface instead of typical microvilli. Their unique function is transepithelial transport. They actively sample antigens like bacteria, viruses, or particles from the lumen and transport them across the cell to underlying dendritic cells and lymphocytes within the Peyer's patch.
So they're like scouts, bringing intel to the immune system.
Exactly. They initiate mucosal immune responses. They're a critical link between the lumen environment and the organized lymphoid tissue beneath.
Are there major differences between the duodenum, jejunum, and ileum?
Some key histological markers. The duodenum is unique because its submucosa contains large, coiled, tubular glands called duodenal glands or Brunner's glands.
What do they secrete?
A distinctly alkaline mucus, pH 8.1 to 9.3, rich in bicarbonate. This is crucial for neutralizing the acidic chyme arriving from the stomach, protecting the duodenal lining, and creating the optimal pH environment for pancreatic digestive enzymes to work.
And the ileum specialty?
The ileum is characterized by the presence of those large aggregates of malt in the lamina propria and submucosa, the Peyer's patches. Villi also tend to become shorter and more finger-like compared to the leaf-like villi of the duodenum or jejunum.
This whole system is so intricate. What happens when it breaks down, like in celiac disease?
In celiac disease, an autoimmune reaction to gluten proteins damages or destroys the villi, especially in the duodenum and jejunum. This drastically reduces that 200 square meter absorptive surface area, leading to severe malabsorption of nutrients, diarrhea, weight loss, and other complications.
And Crohn's disease?
Crohn's disease is a chronic inflammatory bowel disease that often affects the terminal ileum and colon, but can occur anywhere. It's characterized by transmural inflammation, meaning it affects all layers of the tract wall, leading to pain, diarrhea, strictures, fistulas, and malabsorption due to chronic inflammation and tissue damage.
Okay, let's move into the final stretch. The large intestine. Shorter than the small intestine, but wider. Includes the cecum, appendix, colon, ascending, transverse, descending, sigmoid, and rectum. Functionally, the game changes here, right? Less nutrient absorption.
Much less. The primary jobs now are absorbing water and electrolytes from the remaining indigestible food matter and forming and storing feces prior to elimination.
The first major histological difference you notice compared to the small intestine.
No villi. The absorptive surface area is drastically reduced. The mucosal surface is relatively flat, but it's penetrated by numerous straight, tightly packed tubular intestinal glands or crypts, which are longer and straighter than those in the small intestine.
And the outer wall has those pouches.
Yes. The wall is puckered into sacs called haustra. This isn't a mucosal feature, though. It's due to the muscularis externa.
Ah, the muscle layer is different here, too.
Very distinctively. The inner circular layer is continuous, but the outer longitudinal layer is incomplete. It's gathered into 3 separate, narrow, thick bands spaced equally around the colon. These are the teniae coli.
And they cause the haustra.
Yes, the tonic contraction of the teniae coli bunches up the colon wall between them, forming the haustra. These contractions also contribute to the mixing and slow propulsion of feces, a process called haustral churning.
Back to the mucosa. If there are no villi, what are the main cell types in the glands?
Still dominated by absorptive cells, now often called colonocytes. They are columnar, have irregular microvilli, much shorter than in the small intestine, and their main function is absorbing water and electrolytes, driven by active sodium transport. You often see dilated spaces between these cells, indicating active fluid transport.
And the other major cell type seems like lubrication would be important here.
Crucial. Goblet cells become dramatically more numerous as you move from the cecum towards the rectum. They produce vast amounts of mucus to lubricate the increasingly solidifying feces and protect the lining.
Still need immune defense.
Absolutely. The large intestine has an enormous bacterial population, the gut microbiome, so the lamina propria remains rich in lymphocytes and lymphoid nodules, MLT.
You mentioned the teniae coli structure being a potential weakness, leading to diverticula.
Yes, diverticulosis is common, especially in older adults in Western societies. It involves small herniations or outpouchings of the mucosa and submucosa through weak spots in the muscularis externa, often where blood vessels penetrate, usually located between the teniae coli.
What causes them?
Thought to be related to structural weakness in the colon wall, combined with high interluminal pressure, often associated with low-fiber diets and constipation. If these diverticula become inflamed or infected, that's diverticulitis, which can be painful and serious.
Okay, the very final segment. The anal canal, only about 3-4 centimeters long. What defines this zone?
The major transition is again the epithelium. At the rectoanal junction, the simple columnar epithelium, typical of the rectum, abruptly changes to a protective stratified squamous epithelium. Initially non-keratinized, it becomes keratinized further down towards the anus, continuous with the skin.
Any other structural features?
The mucosa and submucosa form longitudinal ridges called anal columns. The areas between these columns contain networks of veins, the internal hemorrhoidal plexus.
And control over defecation relies on sphincters here.
2 critical sphincters. The internal anal sphincter is simply a thickening of the inner circular layer of the smooth muscle in the muscularis externa. It's involuntary.
So it's usually contracted.
Yes, maintaining continence unconsciously. Then, surrounding this, you have the external anal sphincter, which is composed of striated muscle part of the pelvic floor musculature. This one is under voluntary control.
allowing conscious control over defecation.
Precisely. Relaxation of both sphincters is required.
And issues with those veins in the anal columns lead to.
Hemorrhoids. If the veins in the submucosal hemorrhoidal plexus become dilated and swollen, often due to increased pressure from straining during defecation, constipation, pregnancy, or prolonged sitting, they can bulge, bleed, and become painful. So we've covered a tremendous amount of ground. From the stratified squamous barriers at the entrance, through the complex secretory machinery and unique three-layered muscle of the stomach, the incredible absorptive architecture of the small intestine with its triple folding strategy, the water reclaiming function and distinct motility of the large intestine, right down to the final sphincters. Understanding these histological and cellular details, the shifts in epithelia, the specific glands, the role of the enteric nervous system, and MALT, this really provides that mastery-level view of the GI tract.
It truly is an amazing system, especially its capacity for constant regeneration and repair. Which brings us to a final provocative thought, circling back to something we touched on earlier, the teeth, specifically enamel.
Ah, yes, the ameloblasts.
Right. We established that ameloblasts, the cells derived from ectoderm that create enamel, the hardest substance in the body, are completely lost before the tooth even erupts. That structure, once formed, is permanent but fundamentally irreparable by the body's own cellular machinery.
A stark contrast to, say, the lining of the small intestine we just discussed. That entire complex epithelial layer, facing constant chemical and mechanical stress, is completely replaced every few days thanks to those stem cells in the crypts. An incredibly dynamic repair cycle.
So here's the thought for you, the listener, to ponder. What does this fundamental developmental difference tell us about evolutionary priorities? You have one critical digestive structure, enamel, designed for extreme hardness but with zero intrinsic cellular repair capacity. And then you have the rest of the GI barrier, designed for complex function but with an astonishingly rapid and continuous mechanism for cellular replacement and healing.
Does the body prioritize maintaining the function of the barrier, the absorption, the gatekeeping, even if it means constant cellular turnover, over maintaining the absolute structural integrity of its initial processing tools, the teeth?
Function versus structure, rapid repair versus permanent design. Something to think about, reflecting on the incredible resilience and adaptability built into the system right down to the cellular level.